Yes, myself, Latika Chavan. I am going assistant professor from Department of Biotechnology. Today we will discuss basic concepts of genetics. So basically what is genetics? It is a branch of biology which concern with study of genes, genetic variation and heredity in organisms. What are genes? Basically it is a segment of DNA which carries information for a particular trait to appear. DNA, which is deoxyribonucleic acid, which is the hereditary material in humans as well as in almost other organisms which are found on the earth. If we see the basic branch of genetics, it is having two concepts. One is heredity and another is variation. So whenever we are saying heredity, it is transmission of characters from one generation to next, and variations are the differences in individuals which are appearing from one organism to another organism. So first, who is the father of genetics, who is a George Mendel. He has published his work in 1965. Initially, the term was a factor, and later it was recovered or discovered by genes. So now we are not using a term factors, we are using genes for transmission of characters from one generation to next. There are 7 contrasting characters in a model organism which was used for the Mendelian genetics, which is a sweet pea. The scientific name was Pisum sativum, and there are few characteristics, contrasting characteristics, as well as you can say that the plant is bisexual. It is self-pollinating. That means the anther and the ovule can fertilize and produce a zygote which develop into a new plant. Easily these plants are cross-pollinated, and there are few physical characteristics which are very easily recognizable and you can study it easily. The lifespan of the plant is very short and the plants are easy to maintain. So if we observe the diagram of a sweet pea flower, it is a bisexual. It is having both female as well as male reproductive organ. It can be easily cross-pollinated to study the characters which will appear into the next generation. These are the 7 contrasting characters which include what is appearance of seed, what is color of a seed, color of flower, the pod shape as well as pod color, what is position of flower, and what is height of the plant, whether it is a tall in height or dwarf in height. If you observe these 7, there are only 2 contrasting characteristics, means either the plant is tall in height or the plant is dwarf in height. This means it is very easy to identify that a plant is having a dominant characteristic which will appear in every generation, or it may be recessive characteristic which is not predominant in population but it is appearing in F2. So the first concept we will study, which is a trait. So what is trait? It is a characteristic which will be there in every individual or in organism. It is a hereditary trait, means it is transmitted from one generation to next, and it is also called as a character. So if you observe the images which are shown on a slide, whether you are righty or you are lefty, that is a trait. Whether you are able to roll your tongue, that is also a characteristic. What is color of eyes, whether it is blue in color, gray in color, or black, dark brown, that is also the trait which we are carrying because of the genes which are present in us. So observable characteristics which are offered in human beings. For example, we are having attached or free earlobe. It may be a appearance of dimple, it may be a widow peak, it may be the curly or straight appearance of hairs. If you observe the diagram, there are several kinds or possibilities of curlness of hairs which are originally there because of the genes which are there in that organism. If the second concept is true breeding. What is meaning of true breeding? The organism, if you do a cross between the two true breeding species, generation to generation, similar type of organisms will be produced. For example, here a cross is shown between the individual who is AA and the second individual which is AA, means both are homozygous dominant. In F1, which is a first filial generation, you are going to get AA genotype only, which is homozygous dominant. In F2 also the generation will remain same. That means generation to generation the organisms are going to produce similar kind of offsprings, that is called as true breeding concept. Alleles. that are the different forms of gene. So as I have told you, the three concepts, homozygous, heterozygous and homozygous recessive. So the two chromosomes are shown with the alleles, that is a specific location, and capital A and small a is written. Capital A signifies it is a dominant allele, and a small a, as it is a small letter, it specified it is a recessive allele. So there are three conditions, whether it is a heterozygous, homozygous or homozygous recessive, that we will study in detail in next 2-3 slides. Two more concepts are genotype and phenotype. What is genotype? It is a genetic constitution of an organism. So basically, if you want to show that a plant is tall in height, we will write a genotype as capital T, or we may write it as a capital T small t if it is a heterozygous condition. Phenotype is a physical manifestation of the genotype which is there in an organism, plus it is an interaction between the environment where the organism is growing or living. So phenotype is basically produced with the combination of the genes which are there in an organism, plus the environment also plays role in the expression of genes. So here I have shown you a diagram where a round appearance of P and a wrinkled appearance of V, that is because of the appearance of allele which is there in that sweet pea. So as I have told you, there are two conditions. Either it is your alleles are present in homozygous condition, homo means same, both the alleles are same. If it is dominant, that condition is called as homozygous dominant, and if both alleles are recessive, that is called as homozygous recessive. Second condition is a heterozygous, where one allele is dominant and second allele is recessive, that is a heterozygous condition. Two more concepts which are very easy to understand. I have shown you a chromosome, a specific place where our gene of interest is located, which is shown in a pink color or in a white color in subsequent chromosome. That is called as a locus, a specific place where our gene of interest is located. If an organism is diploid, the genes are present, or your homologous sets of chromosomes are present twice. And if organism is haploid in nature, a single set of chromosome is present, that is called as N. The zygote which is produced in an organism which are sexually reproducing, where they will be producing a zygote which is produced by a fusion of male and female gamete. For example, here what is shown is a case of diploid versus a haploid. If you consider, for example, humans, there are 46 chromosomes, out of which 23 pairs of chromosomes are there, 22 autosomes and one sex chromosome. And if you consider a human female gamete, so that is an egg which carries 22 autosomes and only one sex chromosome, that is your X. So 23 chromosomes, that means every chromosome is present single, that is called as a haploid stage, and all somatic cells, they are present in diploid state. Whenever we are crossing, so whatever Mendel has done, they have crossed several breeds and see what is the next generation or the characteristics which appears in the next generation. So first they have done a cross. So what is cross? It is a mating between two individuals. That may be a insects, that may be a plants, that may be a humans. So in case of mating, there are two concepts. First is monohybrid cross and second is dihybrids. Also, although there are possibilities of a trihybrid or a tetrahybrid condition, we will discuss today the two concepts, what is monohybrid and what is dihybrid cross. Meaning of mono is single, means at a time while doing a cross, the consideration is single trait is considered. For example, if you do a cross between a plant which is tall in height and a plant which is dwarf in height, that is an example of monohybrid. At a time you have considered only one trait, that is height of a plant. Dihybrid cross means a cross where at a time two contrasting characteristics are considered. For example, a plant which is tall in height plus a plant which is producing round appearance of a seed is crossed with a recessive plant which is dwarf in height and which is producing a wrinkled seed. So you have considered two traits. One is what is height of a plant and second what is appearance of seed. Whenever we are writing a cross, there are several labels that has to be written or considering while representing a cross. So first, always a parents, and parents are written with a P, capital P generation or parental generation. Usually in genetics, female parent is written first and then it is crossed with a male parent. So on left hand side, you will write a female parent crossed with a male parent. Production of gametes, fusion of gametes, and next generation will be the first filial generation. And if you do a crossing over between F1 and F1, or maybe a crossing over between a recessive parent and the parental one, then you will get a F2 generation, that is called as second filial generation. These are the ways by which we can show a cross, that is a smooth seeded plant is crossed with a wrinkled seeded plant, and F1 generation will be produced, which is having a smooth seeds. So although here the open pod is shown, just to observe how the seed pattern is there, whether they are smooth in appearance or whether they are wrinkled in appearance. So if you observe all the seeds which are visible in a pod, they are having a smooth appearance. That indicates this trait, the one which is expressed in the F1 generation, is a smooth, and that is a dominant trait. If we cross a F1 with a F1, we are going to get a ratio of 3:1, means approximately 75% dominant trait and 25% recessive. Approx means if you do, for example, thousands seed analysis, approximately 750 will be the smooth and approx 25 will be the wrinkled appearance of a seed. How to show a cross by using a Punnett square. So whenever we are doing either a monohybrid cross or a dihybrid cross, female gametes are written on left hand side, male gametes are written on the right hand side. Their random combination is shown, and below the genotype, visually we write what is its phenotype. For example, here tall is crossed with a dwarf plant. The gametes are capital T and small t. I will explain you what is law of segregation which is applied over here. The F1 generation is capital T small t. Again here one another law which is applied is a law of dominance, where wherever dominant allele is there, it will get expressed, and you will get the expression of dominant one only in the F1 generation if it is following a Mendelian law. And next is crossing of F1 with F1 to produce a F2 generation in 3:1 ratio. This is a way by which we can show a dihybrid cross where at a time two contrasting characters are considered. First, what is color of a seed, and second, what is appearance of a seed. So color of seed is yellow and appearance is round for a first parent, and for second the appearance is wrinkled and color is green. F1 is shown, and here if you observe 16 possible combinations are shown in a F2 generation, and again on the basis of law of dominance, whether that seed is yellow in appearance or green in appearance will be decided based upon whether it carries a capital Y or small y, that is a dominant allele or a recessive allele. Secondly, for the second trait, if capital R is present, that seed will have a round appearance, and if small r is there, that means that seed will have a wrinkled appearance. And dihybrid ratio is always coming 9:3:1 if it follows Mendelian's second law, that is law of independent assortment. Two more concepts, whether your gene of interest or allele of interest is dominant or recessive. So in Mendelian genetics, if you observe, till now I have given you several examples. For example, tall and dwarf, yellow and green, round and wrinkled. All these are either a dominant trait or a recessive trait. If you cross your fingers also, you will get an idea whether your right thumb comes on a left or whether your left thumb comes on a right. So that will tell you whether you are having a dominant trait or a recessive trait. So dominant is always expressed in a homozygous as well as in heterozygous condition. Why? Because single dominant allele is sufficient to mask or suppress the expression of recessive allele. Recessive allele will get expressed only in the absence of dominant allele. So that's why your combination has to be homozygous. Next concept is genome and a gamete. Genome is total amount of genetic material which is present in a single cell or in a whole organism, and gamete is a mature reproductive cell that is specialized for a sexual fusion. I have already told you. So your first law, law of dominance, which tells that whatever traits which are appearing in F1, that is because your dominant traits or dominant allele is going to suppress or mask the expression of recessive allele. If monohybrid cross is there, the ratio will come 3:1 because it is following a law of dominance. Recessive character always get expressed in F2 generation only. Why? Because homozygous recessive condition is not at all possible in F1 generation. It will be there in F2 generation only. So you can take any random example of a monohybrid cross and do a practice and check. In F1 always 100% dominant character will express. For example, breeding between a tall plant and a dwarf plant, 100% tall plants will be there in F1 and 75% tall plant will be there in F2 and 25% dwarf. Same explanation is given. Next is a principle of segregation, which is Mendel's first law. The statement is very important. Two members of gene pairs or alleles segregate or separate from each other during formation of gametes. You already know in case of gamete formation, the process which is involved is meiosis. Mitosis is a process which takes place in a body cells. Meiosis is a process which takes place during gamete formation, where the diploid number is reduced or segregated to half. So here what is result of law of segregation is the half the gametes carry one allele and the another half of the gametes carry another allele from the parental combinations. So every gamete carries only single allele of each gene. Once these gametes fused, F1 generation again will be diploid. So this is a reference which is taken from a Russell standard iGenetics by Russell, where a production of F1 gametes and production of F2 gametes are shown. In F1st cross, what is shown is a parental phenotype which is having a smooth seed crossed with a wrinkled seed. The diploid parental phenotype is smooth versus wrinkled. Genotype is capital S, already you know, homozygous dominant condition crossed with a homozygous recessive condition. Haploid gametes, capital S, and the second one small s. Punnett square will tell you how the 100% smooth seeds will appear in the next generation. If we cross F1 with F1, means capital S small s genotype, that is a smooth bearing plant which is heterozygous in nature, is crossed with another smooth seeded plant, F2 generation will be produced in 3:1 ratio, out of which 25% are homozygous dominant, capital S, 50% are heterozygous dominant, and 25% are homozygous recessive. What it indicates? The genotypic ratio is 1:2:1. 1 capital S as to 2 capital S as to 1 small s. That means 25% only are the recessive characters which will express in F2 generation and 75% dominant characters. The concept of test cross is crossing of F1 with a recessive parent. Just to know whatever height of the plant which is appearing tall to us, but we don't know that whether that plant is homozygous or whether that plant is heterozygous. So to know to whether genotypically whether it is homozygous or heterozygous, what is done is a test cross. In results, if it is showing pure homozygous plant, that means the parents which we have considered are having a dominant trait and both are homozygous. By chance in results, if there is an appearance of recessive traits comes, that indicates the heterozygosity is there in a plants at the genotypic level, but phenotypically that plant is showing appearance of dominant trait because the law of dominance is applied. So if you want to show it diagrammatically, you can show it like this, where a cross between a heterozygous plant and a recessive plant is done to check whether that tall or a smooth bearing plant is smooth or tall because of homozygous or heterozygous condition. The second law, that is Mendel's second law, law of independent assortment, 100% it is applicable to dihybrid cross only, because in a dihybrid cross at a time two contrasting characters are considered. You cannot apply Mendel's second law to a law of segregation. Why? Because in case of monohybrid cross at a time only one contrasting allele is considered. while in independent assortment, two contrasting characters has to be there. Next is whenever we show this trait, smooth yellow seeded plant is crossed with a green wrinkled appearance plant. In F1, we are going to get capital Y, capital R, small r. That is a heterozygous condition which will appear similar to parent 1 phenotypically, but genotypically it is a heterozygous condition, that is capital S, capital Y, small y. So here all four alleles are different. So if you cross F1 with F1, the combinations will be like this, where 16 possible combinations are there. And if you observe the appearance of that seed, there are 9 which are yellow round, there are 3 which are green round, there are 3 which are yellow wrinkled, and there is 1 which is green wrinkled. So if you compare these 4 combinations with the parental ones, there are 2 new combinations which are produced in the F2 generation. That is because of law of independent assortment. So whatever new combinations which are produced in the progeny, that is result of independent assortment, because inheritance of yellow is not dependent on inheritance of green. Inheritance of round is not dependent on inheritance of wrinkled. If it is dependent, new combinations are not possible. So the result is because of law of independent assortment, which is second law. Thank you so much for your attention.